introduce to you one of the, I would say, 3 or 4 emerging trends in biomedical engineering, and that is bio-nanotechnology or nanomedicine, depending on which angle you approach this subject matter. But I just want to get started by asking you, do you know how big nano is, by any chance? Heard of the word nano? How big is a nano? 9. Okay, so what is that? So it's a length scale, mass scale, or what time scale? Okay, so a nanometer is a length scale. Nanosecond is a time scale, depending. So nano is just a number. I would like to give this analogy to people who have never heard of the word nano before. So for those of you who play soccer, nano, it's much smaller than this, right? Soccer ball is roughly of a radius 11 cm. But if you were to take this soccer ball and to blow this up into the size of planet Earth, pretty much you're expanding this by, I would say, 10 million times. So if you just to take this size of a soccer ball, blow it up to planet Earth, that's pretty much what you get, okay? 10 million times, okay? But if I were to take the same order of magnitude, but to work the other way around, shrinking the soccer ball by 10 million times, then I'll get something that's like a nanoparticle, okay? So this nanoparticle image is obtained by scanning electron microscopy, SEM, and you can see that the size of this nanoparticle in this picture is roughly 25 nanometers. So you cannot see it by light microscope, you need an electron microscope to review it. So we are talking about very small objects. But why do we care, okay? So you may have this question, what are we going from here? So I introduce you another length scale. You see there's, what do you call it? A Swedish or Scandinavian symbol right here, letter. Any of you know what this is? The A with the circle up there. What is it called? Angstrom? So how long is an angstrom? How big is angstrom? Yeah, 1/10 of 1 nanometer. So if this scale bar refers to 5 angstrom, this means 0.5 nanometers. So even though we think that nanometer is very small as compared to what our naked eye can resolve, if you were to compare 1 nanometer to the fundamental building blocks of life, of nature, like water molecules, glucose, amino acid, a nano is pretty big actually. Okay? If you say a nanoparticle that measures around between 1 and 100 nanometers in size, in diameter, you can pretty much put a lot of these kind of things inside that nanoparticle to form a huge like robot or nanobot. Think about it, right? Even though a nanoparticle is much smaller than what we can see, it's still considered much bigger than these fundamental building blocks of life. That's all I'm trying to say, okay? Next. So just want to give you guys this length scale to bring it to more clarity. Sure, you have seen a red blood cell. How big is a red blood cell? Anybody? Yeah, it's 7 microns. So micron is, this is a micron, or micrometer, depending on how you say it. This mu is pretty much 10 to the power minus 6. So 1 micron is what? 1,000 nanometers. And the red blood cell is right here, 7 microns, so 7,000 nanometers in diameter. Okay. Then bacteria is slightly smaller. This arrow is pointing to the resolution limit of a typical light microscope. Now, of course, with advances on the optics, you can even go push farther the resolution. And I think maybe other speakers in this course in biophotonics may give you more examples. But this is just showing you for typical classical light microscope, it's around maybe 200 nanometers or something. That's your light microscope. It's defined by your, has something to do with the wavelength of the light. And then if you go smaller and smaller for ribosomes, which actually assist in protein translation, hemoglobin forms the basis of, red blood cell, but it carries the oxygen, right? Glucose, and even carbon-carbon bond. You can see, this length scale tells you that nanoparticles here. That's why my title is kind of alluding to you the concept of assembly. If there's a way we can actually assemble these fundamental building blocks to around this regime, you can do a lot of engineering here to see what kind of biomolecules you can pack inside a nanoparticle to do the job of medicine. Okay, so these are some kind of pictures that I gathered from the online website because I think this nano business is getting kind of hot in Hong Kong. I'm sure you have watched a TV commercial, you know, the nano detergent. Yeah, they can break down your debris, or break down your oil, your grease into something that's nanoscopic scale. That's how the one of the TV commercials is selling the detergent products. What's this thing to the top left-hand corner? What's this? Sunscreen. Now sunscreen, what does sunscreen do? It blocks off the light, right? So that's why the light cannot actually burn your skin. And the reason that it works is that there are some titanium oxide particles that are white in color, deposited on the surface of skin, such that you can actually reduce absorption. So that's why that particles, if you want to be safe, got to be bigger than the size that will allow penetration into the skin. Otherwise, why would you want to smear particles that can penetrate through the skin if it's not good, right? You just want it to couple on the surface. So you want that particle to be bigger than permeation limit, which is around 20 nanometers. Okay. What's this? Anybody? A patch? What would a patch do? What? Contraceptive is one. Also, nicotine is another one, like to get rid of smoking. But your parents, my parents at least, would like to have these patches to deliver some kind of muscle soothing molecules for bones, right? Some ancient Chinese therapy. You buy a patch like this. Oh, very soothing. Okay. Okay. Any female audience in the female audience, my mom. My cousins would convince their husbands or boyfriends, can you buy me this mask please, this collagen enriched mask that allow add suppleness to the skin and moisture and whitening effect to the skin. So collagen is actually protein, it's a nanoscopic scale protein. And you embed this kind of protein into this hydrogel that forms the basis, structural basis of the hydrogel of the mask. Then you can have some kind of protein swings slowly going through the skin, right? So that's one application of nanomaterials. This is in Chinese, I'm very sorry, but it does say nano perm, nano curls, nano perm. So you can actually get a nano perm from this neighboring barber shop right next to my place in my neighborhood. It's around $200. All right, so this nano concept is all around you. Okay, it's ubiquitous in consumer products. But I do want to take a historical route to go back to where all nano started from. Because shown here are actually some kind of pre-modern architectural goodies, that one can find back in Europe. You see on the top left-hand corner, there's this vase, right? This vase is kind of red in color. This redness actually, people found out that they actually are gold nanoparticles. They're gold nanoparticles that naturally display a red color due to their interaction with light. So that's why you can see that but then people back then made this vase without knowing that they are nanoparticles. It just so happened that the modern scientific analysis proved that they were actually gold nanoparticles. The second picture right there, this is showing the very famous world-renowned Notre Dame Cathedral in Paris, which I just happened to visit a few weeks ago. You see the window panes right here. Those are all if you see the red stuff, that's all gold nanoparticles right there in the window pane. So actually found in your furniture, found in your architecture long time ago already. So this thing has been around us for a long time. But really the first scientist who actually carved a niche in this nano industry back in a long time is in Britain by the name of Michael Faraday. Michael Faraday was the first one who actually discovered colloidal gold is actually like this, truly discovered colloidal ruby gold, showing that the gold particles under certain lighting conditions have different colors. Okay. So, and this different color ultimately traces back to this fundamental physical properties that is different than the bulk properties at a nanoscale. What I mean to say is that what's the color of gold, right? Bulk gold. Obviously it's gold color. And then needless to say, but the first thing for you to take home is that your nanoparticles are actually not gold in color at a nanoscale. They actually look like burgundy. They're wine color, like that. Okay, so this is actually how gold nanoparticles look like in color. So the nanoscale of the gold renders its interaction with light completely different, such that the gold nanoparticles attain a color of red instead of the gold color. This is, okay, FYI. And there are more and more recent advances. For example, the transistor, how we can actually build transistor in nanoscopic scale, how we can build TEMs, that's in the early 30s, late 30s, 40s. IC circuit, we have the first concept, basically trying to allow you to pack all these circuits in nano level. And closer to our contemporary times, you know the very famous Moore's law? Yeah, how you can actually embed more and more circuits in a given space. You can basically keep shrinking the size of the circuit, right? And also the buckyball. Have you seen this buckyball before, or fullerene? Yes, that's basically a Nobel Prize awarded for the discovery of carbon atoms that join together, 60 of them join together to form this nanoparticle called fullerene, or buckyball, okay? So this C60. That was 1980, blah, blah, forgot, here, right here, in the 80s, okay? And there's another type of nanoparticle called the quantum dots, which we'll talk about in the later part of this lecture. They are naturally fluorescent particles, the semiconductor particles, the inorganic particles that fluoresce. So they give off interesting fluorescent properties that really help our biomedical investigations. What's this? Carbon nanotube. So instead of a ball sphere, you can actually form a tube. I think it's a Japanese scientist by the name of, where is it? Sumio Ijima discovered this structure. They can actually grow this structure to form like this. So 80s is very, very interesting by the discovery and the synthesis and engineering of these so-called nanomaterials. Not only we can discover them in nature, we can start making them. This is really the burgeoning of the nano era is when scientists, material scientists and chemists have developed mechanisms to actually control and predict the morphology and the size and shape of all these different structures they can make. Now you can only have these structures, then you can start doing engineering to maybe you can do medical applications, because you need to have the structure first and then you think about what to do with that structure. 80s is really the heyday of the structural development. Shown here in the 90s, scientists by the name of Kresge from Mobil, that's a very famous petroleum company in the US, discovered there's this structure called nanostructured mesoporous materials, that these structures have these very fine structures, elements that with gaps inside. So if you have some nanoscope with these nanostructure with these kind of metals, they can have kind of gaps of all the size inside this structure. They can do catalytic functions. That means they can speed up chemical reactions. And this is a very useful property for the petroleum engineering industry. So this is to say that because of the pore size, they can selectively control for certain molecules to go in to react and block certain molecules that are too big that do not go in. So you can endow this concept of selectivity. What molecules I want to react, what molecules I do not want to react by this structure. Okay. So this was really back in the like the good old 20th century. I've recently had the chance to read this paper that was presented recently. You can see that these are the one of the graphs showing the growth of nanoscience in terms of academic publications. You can see different countries are doing very well, putting government level, academic level, industrial level, putting their energies and resources into developing nanoscience. You can see actually China is actually picking up very, very fast. Actually, now it's actually the world leading in terms of their publishing. And if this is the total number of publications, the right panel is showing to you that the top journals, publications, the top journals related to nanoscience and nanotechnology, China has actually overtaken the US. So this is really recent, okay? So China is really catching up. And so what do these nano people do? What kind of structures do they do? I featured the nanoparticles, right? Which we'll get back to it very soon. But there are actually another, the nanostructure material was the MCM41, which is the catalytic mesoporous material for catalysis, right? Developed in 1992. Nanosheet, what would be nanosheet? For example, graphene. Here we have graphene, right? Carbon atoms, they're kind of like planar, one atom thick. That's one type of nanosheet, okay? Nano device, nanoporous material, nanofilm, nanowire. Most famously, engineered by Professor Pei Dongyang, for example. Nanotubes, carbon nanotubes, for example. So different industries, different countries, when they publish, they go into different types of nanomaterials. This is just a survey, okay, of what type of nanostructures we talk about. Now, once you make these structures, what on earth do you use these structures for? Because you can't just keep making structures and do nothing, right? It turns out that many of them actually do go into semiconductor electronics, just making different structures. See, this is where we sit here. Nanomedicine is here. Paintings and coatings, those are for other processes. But since this particular paper talks about the development of China in nanoscience in the past 2 decades, there's this statement which I just copied from the from the textbook, from this paper, this white paper actually, white paper. They say that China has a clear leading edge in catalytic research. It's the most popular research area in terms of nanoscience in China. Then you ask, what's the second? The second is actually, nanomedicine. So nanomedicine is the second most popular type of research nano research that's going on in China. So you can see this is a very interesting graph that I found in this white paper, in terms of the different applications and in terms of how different countries are doing in terms of the contribution to this field. For example, you can see electronics, South Korea is doing a leading job. You can look at your Samsung, you got LG, right? Your OLED, your TV screens, So that's Samsung right here. Different countries have the different kind of contributions. Okay, that's just something interesting for you to take a look. But the point I want to copy in direct relation to this lecture is that it's very funny how this is actually from the editors. They were saying China is picking up in the ability to actually control these the synthesis of these architectures. They have different ways to do it. But this might come as a surprise to some because China's life science, that is biological research output, like if you're from SLS or SBS, they kind of lag behind compared to the other parts of the world. So this is really the BME part of this is coming that may potentially affect China, in my opinion, because you can leverage your engineering expertise in material science and chemical engineering to boost the development in life science. Because at the end of the day, we need very advanced, drug carriers, diagnostics to improve, to help doctors, do the job, to help save patients, right? So this is something that China and nature, they think that China can leverage its strength in this particular field. Okay, so this is something for you to kind of pay attention to. All right, so in order to make these nanomedicine structures for medicine purposes, nanomaterials for medicine purposes, what scientists and material science do is to actually combine. You can have organic or inorganic materials, but with fundamental building blocks of life. And there are actually 4 types of building blocks. And in my course that is happening right after this one today, 4510, if you get a chance to take it 2 years later, we'll be explicitly going after these 4 different types of biomolecules, ranging from nucleic acids, which makes your DNA, proteins, which you're improving your peptides and amino acids for your structural and catalytic function, lipids that really source energy and makes your cell membrane, right? And also sugars, right, for your energy. So we'll be doing this in another course, but I'm just saying that to build bio-nanomaterials for medical applications, you gotta be actually using any of these type, or even all, or some, depending on your purpose. Engineering how to package these molecules into the nano-sized structure so that you can do your thing. Okay, you got me? Yes. All right. So this is the key point here. Now, just want to inspire you guys what you can do with these nanostructures. Why can we use here? So this here is a very interesting diagnostic kit that uses nanoparticles. What you can see here is actually a glass slide. And on the surface of a glass slide, it turns out that you can grow some DNA. You can grow a short piece of DNA right here, which have this particular sequence. Now the point is that if you are to combine another gold nanoparticle, so this is nano here, this is nano. You have a gold nanoparticle that contains DNA sequences that with this sequence. So this sequence and that sequence, they're not the same sequences. But why are they being placed in proximity? It's because the idea is that the combination of the use of this glass slide and the gold nanoparticle allows for the detection of this particular longer strand of DNA that is sitting on top of it. Once they come together, there's this hydrogen bonding to form the Rosen and Crick, right, duplex. Then this gold nanoparticle it's like this, right? So you have this glass slide sitting here. You flow in your target DNA and flow in your gold nanoparticle. Then your gold nanoparticle can be duplexed and then kind of levitates on top of this gold glass surface. And as a result, the gold being burgundy in color, this part of the glass slide will then appear to be burgundy red. And the burgundy redness of this will then reflect how much actually your gold nanoparticles may bound, which in turn suggests to you how much of the target DNA you captured. So what is this tool for? It's a diagnostic tool for target DNA. It could be your virus DNA, your cancer DNA, what have you. So basically this is telling you, by doing this, you can actually, the detection limit can be pushed down all the way to 50 femtomolar, which is, femto is like what? 10 to the power minus 15? Pico is minus 12, right? So nano, you go like this. Milli, and then micro, and then nano, and then pico, and then femto. So this is, I think, minus 15, yeah? So that's pretty low. We can actually go down already in year 2000. So this is why there's quite a lot of amazement in this nanomaterials community. This particular example has nothing to do with the nanoparticles now, but it has to do with something nanomechanical interactions. What they do here is that they again, find some way to make this chip. This chip contains some gold on the surface of silicon nitride. And the point is that on the surface of these chips, you coat some receptors. Now these receptors can be antibodies, can be some receptors, what have you. But then they would be specifically recognizing the type of biological molecule that you want to detect. And the idea is that if you flow in a fluid that contains this target molecule, this diamond-shaped target molecule would be specifically recognized by this purple-shaped receptor, and as a result of the increase in the binding, this entire chip will feel the increase in weight, and ultimately this cantilever will then deflect downwards. This deflection will then in turn tell you how much particles are being bound onto the surface. That can tell you the concentration of this particular analyte in that fluid. So this is why nanotechnology is helping us. This thing is all nano. This is the SCM of this particular picture, right? This is all nanometers in scale. The type of force that you're producing is also nanomechanical in magnitude. So that's why this is also a different type of nanopart, a different type of nanotechnology application for medicine now. Okay. What else is interesting? This is interesting too. It turns out that for your cancer cells and your normal cells, it would be a good way that you can actually tell apart if you have a mixed cell population. What actually are cancer cells? What actually are normal cells? It turns out that, for example, this is what a cancer cell looks like. This is what a normal cell will look like. The cancer cell got balled up in shape in this particular example. You can visually tell it, but wouldn't it be nice if you have an AFM, atomic force microscope, which is, by the way, a very fundamental piece of invention for nanoscience, can detect the strength, or depending on, they will detect the stiffness of the cell membrane. It turns out that this very interesting clinical study shows that if you have a cancer cell, the stiffness of the cancer cell is actually 70% softer than a normal cell. So imagine you have a tip that goes like this. It just goes around and around the sample and kind of tapping onto the cell membrane surface, and then this AFM will then record to you the amount of force that is required to push to indent the particular cell. And you can see that cancer cell happens to be 70% softer than a typical normal cell. So that's interesting, right? So now, all these three examples, what I'm showing to you, are examples that are just ex vivo, so they're not really inside the body, but they are just taken with solutions of analytes or even cells that we just put into test tubes or in this testing platform to see how they look like. Okay, so this is one branch of bio-nanotechnology or nanomedicine. Next, I'm going to tell you that there are the second branch of nanomedicine or bio-nanotechnology entails actually the delivery of nanoparticles into a cell or into an embryo. Why do we want to do that? Well, in this case, for example, quantum dots, as I alluded before, are naturally fluorescent particles. In this case, the scientists actually added a test tube with the quantum dots and this fluorescent nanoparticle inject into one of the cell, and you can actually, using microscopy, to show how these nanoparticles actually diffuse from one cell to the other. This just tells you that it's very cool. You can actually see the life. So the embryo is still alive. Okay, life tracking of the movement of the nanoparticles from cell to cell. That will add a lot of you can get a lot of information, right? The relative size of the cell compared to the others in the embryo, the speed of the diffusion, as a function of temperature. All these things are very interesting parameters that will be made possible by this kind of fluorescent particle that allows for tracking by microscopy. Okay, so nanoparticles are actually playing a good role here because you can allow tracking. Another type is that instead of doing this tracking in the embryo, we do this in the animal now. So this really goes down to the concept of cancer detection. So I'm sure you know that cancer represents a very huge economic burden. Many of our biomedical engineers are trying to find different ways to work with doctors to come up with innovative ideas to combat cancer. One of the more conventional way of thinking in our field or in the field of cancer nanotechnology, is the use of nanoparticles that look like this. So shown here, you see that it's actually a nanoparticle in the middle. This could be magnetic if this were iron oxide, or it could be fluorescent if this were a quantum dot, right? It doesn't matter what this particle is. It depends on what kind of imaging capability you want to endow to this nanostructure. Attached to the surface of this nanoparticle, you can see these are dextrin. Dextrin is sugar. It's one type of sugar. So these are sugar strands that are being attached to the surface. And at the end of the sugar linkages, you can see this RGD. So this is actually a peptide now. So a short piece of peptide that could target certain receptors. So that looks like this sickle shape. This receptor, this is a cartoon of a receptor around 6 nanometers in size. Is overexpressed. That means that cancer cells have a higher expression of these receptors than normal cells. This really, I'm not sure, have you heard of the word targeted therapy? Okay, so targeted therapy means that if there's a way if we can selectively deliver drugs or image cells that are more cancerous than the other types of cells, then potentially we can actually do less harm. to the cells that you don't want to hurt with the drug. You're only going after the cancer cells, right? So what this idea is that this idea presented in this slide is the fabrication of this nanostructure. You see, overall structure is around 30 nanometers in size, this whole thing. So now you can see the texture to it, right? You combine the coating that make the particle stable in fluid. You put on the targeting elements towards the end that you can recognize different receptors on the cell membrane. Sugars, peptides, iron oxide particle. Wow, you see, building blocks. Can you see the concept now? You're building off of different types of classes of biomolecules. Sugars, peptides, and then with the functional particle, the core in the middle for imaging, for a particular application. So this is really called Lego building now. You're building something up to building a very interesting structure to do your purpose. Now, turns out that people have done this in the middle 2000s. Instead of so for the core, they actually put in a fluorescent particle called a quantum dot, which you've just seen in the previous example. Here, what they have done is that they actually put some of these different type of targeting ligands to target the cancer cell receptors in an animal. So what they've done is that they actually planted some tumors subcutaneously, so right beneath the skin. Then they injected these quantum dots that contain these targeting ligands. And the idea is that can these particles find their way to the tumor? And if they do find their way to the tumor, that means that the tumor will be lit up fluorescently if we were to image them. And that's exactly what they saw. They saw that by doing this complicated signal processing, which, by the way, you will learn from your other instructors in this course, then you can see that the tumors actually light up. This is, by the way, in vivo imaging. In vivo imaging means that the mouse is still alive. I'm not actually killing the mouse first and take out the tumor to analyze. I'm actually doing this live, okay? So this was a very important breakthrough in our field. The ability to actually design nanomaterials that support this analysis by imaging. And by the way, so this is actually at the end of the experiment, they sacrificed the mice, took out the different organs, and actually investigated the distribution of these fluorescent particles. And they saw that these quantum dots can really go into the tumor like this. Okay. So just want to inspire to you what our field has been up to in the past decade or so in terms of nanomedicine. This time, not in a cell or embryo now. This is talking about animal. So you inject the particles into an animal, look for a tumor, and then image. Or then you can tell a doctor, oh, perhaps if I do a surgery right here, I can get rid of it. Okay. So that's the direct clinical implication. I've just given you an example in terms of the fluorescence property of the nanoparticle, but you can easily switch the fluorescent core, nanoparticle core, with a magnetic nanoparticle core. And in that case, in such a case, we'll be doing MRI. Okay, MRI. So you can see this is an interesting picture that I took out from one of the papers, showing you that here is one of your tumors in the brain. If you can actually, this is without the magnetic nanoparticle, but if you were to inject the nanoparticles, magnetic in this case is an iron oxide particle, that will be targeting a particular tumor site, they will look black, okay? And this transition in the intensity from this white-looking region to a black region would then readily signal to the clinician that there's a tumor right here. Please excise it, remove it, okay? You got me? Yes? So this is telling you that why nanoparticles are useful, because it helps the doctor to visualize accurately where the border is, right? Then they can just trace around the border and actually remove it. Yes? Okay, so this is really imaging, what imaging is up to. The third type of application besides imaging is delivering drugs now, finally, okay? So here's the idea. I think I've told you that small molecules, roughly 2 to 3 nanometers in size. The problem is that once you inject them into the body, because they're so small, they'll be peed out, they'll be urinated very quickly. So they do not have time to actually circulate inside the body to look for your organs, because the fast clearance due to renal clearance. Okay, what would be the better way to circumvent this bottleneck, delivery bottleneck, is then to decorate, is to assemble this maybe 100 or 200 molecules like this into this much bigger structure compared to the individual particle. Let's say in this case, they assembled maybe 300 molecules, drop molecules embedded inside a particular bigger nanoparticle that is roughly measuring 30 nanometers in diameter, and to inject this whole thing. And this would, for one, prevent these individual nanoparticles from leaving the body too soon. So if this is not making sense to you, I just want to give you a fun fact in biology. you know, if you go to your bathroom when you take a pee, when you actually look in the urine, they should look clear, right? For healthy, you know, they should look better, look clear. But if you do see that your pee becoming more milky, that's not good. And the milkiness indicates the presence of proteins or aggregated proteins. Aggregated proteins would may then, you know, leave through the kidney to urine because your filtration barrier is not working properly in the kidney. So that's why even larger molecules like antibodies that is larger than 10 nanometers can be peed out, and we don't want. So that's why, going back to this analogy, I want to make sure that my drug molecules will not be prematurely leaving the body. So that's why I want to make this whole thing to be bigger than 10 nanometers, so that they can circulate inside the body for a longer period of time and eventually go to the tumor they want to go to. So this is one of the commonly used delivery principles in our field. But the analogy for you guys is that if you're really into Greek mythology, you know this war, right? The Trojan War, the Trojan horse and the Trojan War, yeah, between Sparta and Troy. You know, you see that each individual soldier will be the drug molecule in this case, and the Trojan horse will be a nanoparticle. Okay, so I hope this message is clear, because what would be the enemy here? Yeah, your physiological barrier, because they'll clear up your particles so soon. Your fluid will clear up your particles so soon. But if you were to package them like this, then you offer each individual particle some protection against premature clearance or degradation in the body, right? Which is very important. Some of the molecules, for example, may not even be water soluble. Let's say some of the small molecules are very hydrophobic. If you were to inject them into the body, they don't dissolve well. So what then you do is that you perhaps can encapsulate them into a more hydrophilic, water-loving polymer to protect them inside, and only release that particular nanoparticle when it arrives at the site of interest, and then release it, and then the drug molecules then diffuse out locally at that point. That's really our hope, okay, in our field. If we can do this, bingo, okay. All right, so one good example in this. One good example of this Trojan horse, if you will, is the liposome. Liposomes could be natively found in the body. People have discovered the existence in 1961. We can also make them too. So, what are liposomes? Has anybody heard of liposomes before? Oh no, this is the first time. First time? Okay, but have you heard of like a cell membrane that has a phospholipid bilayer? Yeah, two sets of. So, do you know why they take the bilayer shape? By the way, because the head groups are hydrophilic, they love water, but the core of the carbon chain groups, they don't like water, so that's why the hydrophobic groups will then aggregate with one another, self-assemble, to minimize their interactions with water, which is surrounding this membrane, right? So this is the idea, why the hydrophobic core groups are being hidden in the middle of the cell membrane bilayer, phospholipid bilayer. So this is the linearized version of cell membrane, but turns out that you can actually make an artificial version of this in a spherical manner, and this is what we call liposome. Liposome is basically a spherical version of a phospholipid bilayer. And they come in different sizes and shapes, smallest of which could go down all the way down to 30 nanometers, which is exactly the shape that we talked about for drug delivery. It turns out that if you have a much bigger particle, they sometimes will go to the liver and spleen. They get uptaken very fast. That very leaving few of them will actually go to the tumor. And once you actually go into the tumor, if your particles are small enough, then they could diffuse through the tumor very much faster than the other bigger particles. Okay, so these are the engineering concerns that if you're really into the field, you will start learning and developing. But they come in different forms and shapes. Okay. Now, this is really one of the first particles being developed. Is a liposome-based doxorubicin conjugate, commercially called Doxo. Have you heard of Doxo? No. So Doxo is basically, you can see here, doxorubicin is an anti-cancer drug. It's a chemotherapeutic. This is your individual soldier now. This is your soldier. Now this soldier is now being packaged into the huge liposome like this. This liposome is then your Trojan horse. And then the idea is that starting from the early 70s or late 70s, people Gabison and Barinholtz started the basic research on liposomal doxorubicin, and then it took them quite a long time. They filed for the first clinical trial in '84, but unfortunately it failed. Very sad. It failed in '87. But they didn't give up. They actually put in some new technology. They introduced a stealth concept by putting in some polymeric strands on the surface to endow with better evasive ability against immune responses. And eventually they actually started Doxo again, finally conducted the first in-human clinical trial in Jerusalem, 1991, and then finally met FDA approval in 1995, and then out for sale in 1996. So Doxil represents the first FDA-approved nanoparticle-based therapeutic, and it is a liposome-based. So it's basically a liposome Trojan horse encapsulating a doxorubicin small molecule drug. Okay. How does it work? Well, you can see here. This is your hydrophobic layer from two. I can go back to the previous slide. You can see that there are actually hydrophobic regions here, and your doxorubicin will then sit comfortably inside this hydrophobic layer because the molecule itself is also very hydrophobic, so they will sit inside the bilayer like this. So this is how the drug is being encapsulated, and then they injected this entire nanoparticle formulation, which by the way looks like has this size, right? This is a cryo-EM. This is an electron microscopy picture of how big the doxil nanoparticle looks like. It's around what, maybe 50 nanometers, 100 nanometers or so. Okay, so this is FDA approved now, and liposomal dox was the first FDA cleared drug, but it turns out that you can actually make good use of the liposomal platform to carry other types of drugs, not necessarily just for doxorubicin. You can also do amphotericin B for fungal infections. You can also do dox, which we covered for ovarian cancer, breast cancer, sarcoma, different other molecules for pain management, hepatitis, meningitis in the brain, like that, influenza. Okay, so it's the delivery platform that is still constant, but the drug that you engineer inside will allow you to serve different types of indications, different diseases. Okay, so this is really the heart of drug delivery. Using nanoparticles as a structure, combining, in this case, what are they? Lipids, right? Liposomes. Lipids, also a fundamental class of molecule, with the drug molecule, doxorubicin, to form this interesting nanostructure to deliver to the tumor, and finally approved for clinical human use. Okay. So just want to share with you some of the work that your TA has done in the audience. So for a long time, people have been using liposomes for a long time, but we really want to understand how many of these, alkyl chains, there is these hydrophobic chains that we need to actually to enter cells. And what Carla, your TA, has done recently is that she found out systematically how you can engineer different amounts of the carbon chains to be attached onto the surface of the nanoparticle, and also the length of the carbon chain to be attached onto the nanoparticle will allow for entry. So shown here are actually pictures of cell pellets. White means that those are natively looking cells. And you remember, gold nanoparticles are red in color. So if you see an increase in red color, that means that more nanoparticles actually gone into the cell. So this tells you that you need to make sure that your carbon chain needs to be at least 12 carbon atoms long in order for you to see any good uptake by the cell. For 18, you can even go to a push. If you go 18, even more. You can see this, red, too red becomes dark like this. Okay. So just a lot of engineering. I just want to keep this slide to you to motivate to you all. This is what engineering people do. We really want to study the effect of different parameters that govern the structure, the function of these nanostructures. To engineer them such that you want them to go in at a certain rate, at a certain amount. You tune them. Where do they go? Okay. This is just some kind of TEM, and by the way, TEM is also very important for nanomaterial science research. Initially, this invented in the 30s. So shown here is actually some gold nanoparticles inside a cell. And if you've never seen a picture of a TEM of a cell before, this is showing you a nucleus of a cell, and this cytosol refers to the cytosol. EX means the extracellular region, that means you're outside of the cell. You can see if you were to actually zoom into different regions of the cell, you can see different quantities of these gold nanoparticles. This left column means no carbon chains attached. This means with carbon chains of 6 carbon atoms attached, 12 carbon atoms attached, and 18 carbon atoms attached. You can see once you actually reach 12 or above, you can see a lot more gold nanoparticles entering the cell. Okay, so you can see the very poignant effect of the carbon length on cellular uptake. Okay, so I just want to end this presentation soon with these two slides. These are slides that I actually use in bio-nano technology 4450, which is another course that I taught before with Professor Aaron Ho. Why do we want to deliver particles into the cell? If we want to do that, how do we engineer the particles so that they can enter the cell more? Well, we just talked about the carbon chain of an alkyl chain of a lipid. Now, what about DNA? What about proteins? What about sugars? What are the design rules? How do we engineer to go into the cell more? Well, interesting question. And the biologists will come into play too. The life scientists, from MBT, also from SBS. How do they go into the cell? Would the pathway that governs the entry be different for different types of cells? Let's say for pancreatic cancer cell versus like a breast cancer cell, would they be different? Different cells take apart with different degree. Would there be anything that's different? What's the biological mechanism that governs the entry of these nanoparticles? We don't know. And once they go into the cell, where do they go? These are just food for thought questions, okay, that really, if properly addressed, will enhance our research so that we can design even better nanoparticles than before. Once they go into the body, wow, you're looking at such a huge design space. They can be huge, they can be positively charged, negatively charged. They could have different types of proteins or lipids, as I mentioned. They could have even different shapes. You know, it turns out that you can make gold nanorods, prisms, cubes, cages, or even slimy worms like polymeric micelles, phyllomicelles. They're different shapes too. And also different material properties. Do you want it to be fluorescent? You want it to be magnetic? You want it to be plasmonic? You want it to be different? Oh, different types of molecules. You can do different things. So yes, the design space is vast. You can pretty much exploit your creativity to engineer your dream nanoparticle to do your particular function. And once they enter the body, where do they go, right? How do they interact with themselves and with the kidney, with the brain, with different organs? How do they go from organ to organ? Once they go into the organ, do they go to this particular tissue type or that particular tissue type? And then inside this particular tissue type, do they enter this particular cell type or not? And then once they enter the cell, do they enter this particular organelle or not? Wow. I don't know, it's a very complex problem that really ties in your expertise in chemistry, chemical engineering, material science, BME, medicine, and obviously I cannot do it all by myself. I just want to give thanks, shout out, thanks to the people, to my PhD students, undergraduate students in my group. So if you're interested in learning more about what we do, feel free to talk to me. After this lecture or even throughout this course while I'm serving as your course coordinator. Just want to thank my funding sources. And I just want to add one more thing. If you want to do iGEM, some of you may want to do iGEM, please talk to me, because you need to, I hope that you can audit this course so that you can pick up some of your basic biology that is required to do well in iGEM. Okay? With that, I can take questions from the floor. Thank you.